Good afternoon, everyone, and thank you for joining us for today's webinar titled The Key Benefits of ddPCR over qPCR, presented by Bio-Rad's Field Application Specialist for the Genomics portfolio, Luke Forbes. We will have the chat function available throughout this presentation, so please feel free to ask any questions you might have, and Luke will address these at the end of this presentation. Thank you so much. Over to you, Luke.
Thanks, Michelle, and thank you everyone for joining. I hope that you found this some of the session at least valuable for you, and yeah, please feel free to ask questions. It doesn't have to be about this particular webinar. Maybe there's something else you want to know or something you've heard. Please feel free to ask in the chat as well. Okay, so to get started, I always like to ask this question when talking about real-time PCR, because anyone that's running real-time PCR knows the pain of some of these things. So do you enjoy preparing and running standard curves? Do you enjoy having poor reproducibility between sort of labs and departments? Do you enjoy having failed experiments due to maybe inhibition, especially if you're working with wastewater? It often happens. And do you enjoy doing sort of complex statistics and statistical data analysis? This is when we're talking gene expression, delta CQs, and all these kind of things. I think the answer to all four of those questions is very likely no. So if that is you, then the great news is for all of those questions, ddPCR is here to help and make your life a lot easier. I'm not going to go into detail on each of those points, because really, obviously, that's the meat of the discussion that we're going to be having this morning. And it's really just going to focus in on some of those points, and we're going to talk through the differences between the technologies, and then we're going to look at papers that have been published comparing them on the various points and seeing where the benefits sort of lie when we compare the two sort of big technologies. So just as a recap, I'm sure that almost all of you in attendance know what PCR is, but it's a good sort of base to build off of, because digital PCR or droplet digital PCR is a type of PCR just like conventional or real-time PCR. So they all use the same technique where you amplify a specific sequence, and that allows people to produce millions of copies of a particular segment of that initial amplicon or a small sample. And obviously, we know that PCR involves cycling at various temperatures, typically three main steps: the denaturation steps, which is the double-stranded DNA templates heated and it breaks into two single strands. Then your annealing stage with your reactionist cool down to allow your primers to bind, which are complementary to your target DNA. And then the specific regions flank the target sequence. And then the extension phase, in which the reaction is raised, the temperature is raised, and your DNA polymerase or your enzyme extends the primers by adding complementary nucleotides, and this obviously synthesizes a complementary strand. And obviously each step effectively amplifies one target sequence, and by repeating these steps multiple times, you get doubling of all your samples with each cycle. And this leads to the sort of exponential amplification of your target sequence. And then obviously you can do downstream analysis on that DNA with cloning, sequencing, detecting specific genetic markers. So really a fundamental sort of application or tool that's become used throughout the genomics and molecular biology sort of fields. Now, when we look at the three generations of PCR, we sort of discuss the first, which is qualitative. We call that conventional or endpoint PCR, in which your answer is just a positive, negative. It's either presence, absence. Is there your target of interest? Has it been amplified? Is it present in your sample or not? And obviously that's the same PCR process. It's just run on a gel to visualize. Then you've got your real-time PCR or qPCR, which is a semi-quantitative relative quantification method, in which you are running the same PCR process. You just have some additional reagents, and you need to use a standard and controls, generally a standard curve, to make sure that your quantity is accurate to another relative sample. And then we've got Droplet Digital PCR, which we like to define as the third generation of PCR, which is an absolute quantification method. And in this method, it's literally counting the copies that you have in your sample. So what is the same across the PCR methods? Well, the same is that the reagents are basically the same. CNTPs, it's your polymerase enzyme, it's got buffers, primers, and templates. That all stays the same, and that's your supermix. Obviously, they vary slightly between the different sort of generations. Things like viscosity, enzyme activity can vary, but the basic makeup is the same. And there's the same thermal cycling process that occurs. So all the methods that are discussed, all three methods make use of the same reagents to a degree and the same cycling profile. However, what differs? Well, in conventional PCR and in ddPCR, we analyze the data at the endpoint. So if you look at that sort of curve taken from a real-time PCR system, you can see at the endpoint is called a non-exponential or the plateau phase. And that's where in conventional and ddPCR, you analyze your results. However, in real-time PCR, it's different. You analyze your data at a CQ, a quantity cycle, and it's that exponential phase where the amount of your PCR product is hopefully doubling between each cycle, remembering that that doubling is because your DNA is splitting into half. But since real-time quantification is based on the relationship between the initial starting template amount and a CQ value obtained during the amplification, an optimal real-time PCR assay is really essential when you're trying to determine quantification. So you need to make sure that your assay is really well optimized in terms of efficiency, and it's not having off-target amplification and things like that, because you're looking at your sample in the middle of the run and you're assuming that it's doubling at that stage. So you need to make sure that it is actually doubling, and that's why real-time PCR can become a little bit complicated. And then again, what differs in detection chemistry. So in conventional PCR, we have no fluorescent dyes to monitor the reaction. Hence, you run a gel at the end of your reaction to determine the presence or absence. However, in both real-time PCR and in the Droplet Digital PCR, we make use of these fluorescent dyes that can monitor your reaction in real time, and ultimately they're useful in determining quantity. So if you think about what we discussed in that slide, you might think, well, obviously ddPCR is kind of the logical approach. It takes what's best in real-time PCR, which is the fluorescent dye used for quantification, as well as what's best in conventional PCR, which is your endpoint detection method, which obviously reduces the effects of inhibition and reaction efficiency is not so important. But what was sort of the limiting factor of digital PCR for a long time? Well, like most things that are sort of happening in modern science, the technology was the limiting factor. So the biggest issue was having sort of accurate partitioning. Accurate partitioning is sort of the most essential part of ddPCR, because your samples all have to be divided into the same sizes. So each partition or droplet has to be the exact same size as the other. And obviously building a method where you're taking a 20 microliter reaction and dividing it into 20,000 smaller volume reactions is obviously very complicated and requires highly precise instruments. So that's why only until about late 2010, 2011 was there really acceptable commercial methods. But in 1999 earlier, there was initial partitioning to sort of divide a sample from 1 into 384-well microplates. But just understanding that sort of understands the building up of conventional PCR, real-time PCR, and then going to ddPCR or Droplet Digital PCR or just digital PCR in essence as well. So this is I'm sure, slide that you might have seen before, and it's sort of defined as, imagine if you're looking at needles in a haystack, and you have a metal detector. Now, if you wave the metal detector over your haystack, it will tell you that there's a needle there, but it can't tell you where the needle is in that haystack, or if it's one needle or 500 needles. If you really want to count the number of needles in that haystack, you're going to take that large stack of hay and you're going to divide it. And in digital PCR, you divide that haystack over around 20,000 times. And in some of those haystacks, there'll be needles and you'll be able to count them quite easy because they'll be separate, and others they won't. Now, I also just want to expand on this a little bit more because obviously we're specifically focusing on real-time PCR versus digital PCR in this instance. So if you think of real-time PCR, think of it as like estimating how many fish are in a lake or in a dam by taking the number of samples over time. So when you do this sort of method, you don't count every single fish in that dam. You kind of track how quickly you can catch fish, maybe, and that's like an amplification curve. And then you compare that to a known lake or a known dam, how many fish are in there, which is your standard curve. So you see, okay, I can catch X amount of fish in this amount of time. That's your amplification curve in real-time PCR. And I know that in this lake, there's 10,000 fish, and it takes me X amount of time to catch them. If I compare the two, I'm getting a relative answer. The lake has about twice as many fish as that lake. So the point when you've caught enough fish to be certain about your accuracy, that's sort of supposed to be your CQ value. Now, in ddPCR, you think of it like dividing that same lake into thousands of sort of small ponds. You partition your sample into thousands of tiny droplets. Each droplet is then counted as it has DNA target or it doesn't, so it's a yes/no result. And you simply count how many ponds contain these fish, and then you don't need to compare between different lakes or different timings of the year, because the result is absolute. You know exactly how many fish are in the dam, so it's 347 lakes or what. So the bottom line is real-time PCR tells you approximately how much by measuring when, and ddPCR tells you exactly how much by counting the individual events. So it removes the sort of estimation by comparison step, giving you a direct count, which means ddPCR is really valuable when looking at something like when using a precise sort of method, or you'll need to know an exact count. Just as a sort of review, because I don't think many people are aware of how the technique works, that digital PCR, the workflow is very similar to real-time PCR or general endpoint PCR. So you prepare your reaction. That's, you've got your template, you've got your super mix, you've got your assay or your primer and probe, or you can just use primers if you choose. The only difference is once that's on a 96-well plate or a standard PCR 96-well plate, you do what's called a droplet generation step, and that's in a specific instrument. And in that instrument, it takes each well of about 20 microliters, and it splits those wells up into around 20,000 droplets each. It only takes about 2 minutes to do a well, so it's not a very long time. And then you can decide between 1 and 96 wells, just like you can in real-time PCR, how many samples you want to run, and then it generates the droplets in each of those wells. Then you move it into a standard thermal cycler, so that's just an endpoint thermal cycler, and you go through the thermal cycling process. And in essence, is what's happening is in each of those little partitions, sort of a mini PCR is taking place. And inside the mini PCRs, either a positive or a negative. And also just keep in mind, because that partitioning effect is occurring, unlike in real-time PCR, it's sort of a bulk sample, your enzyme and your sort of sample can pre-mix together as it heats up and cools down. However, in ddPCR, you need to make sure that you're vortexing fully, because if a droplet is partitioned, what's inside the droplet is never going to change. So if you don't have enzyme or something like that, then obviously that could be an issue. But that's easily overcome by just increasing viscosity of enzyme and then vortexing between your mixing steps. And then finally, after you've amplified your sample in a thermal cycler, you just move it to a droplet reader. And in that droplet reader, it goes to each well. it pulls up the droplets in a well in a single format, a single file, and then it runs them through an LED detector, and that LED detector excites each droplet one at a time, and it sort of determines, if there's this label, FAM, hex, cyber, or EVA green, and it plots them as positive or negative, gives them an amplitude, and with that amplitude, you can determine, does it contain your sample of interest or not? So this is what your digital PCR data looks like for a single target. So each partition, like I said, is interrogated in that reader for a fluorescence signal. So it can be FAM, hex, and it can be more depending on the system. And the fluorescence intensity is plotted on the axis, which are droplets. And then a threshold line is sort of set between to differentiate the droplets where amplification has taken place. So positive for the target of interest, and droplets that are at baseline are negative for your target of interest. And then obviously the positive and negative partitions are counted, and then that determines the concentration and the target of interest. Keep in mind that it is just a positive negative scoring, so that makes it digital 0 and 1. But it also it fits a statistic model just to determine if there's a likelihood of a double positive occurring, which can happen. And that follows what's called a Poisson distribution. I'm not going to discuss that in today's webinar, but you can do some research, or if you have some questions, you're more than welcome to ask me. Just to note that this is a 1D target, so it's just a single target versus a negative. So let's say FAM versus a negative, not a dual label or something like that. So you're only looking at one target per time. This is what your data will look like if you start to look at it in a 2D multi-target sort of assay. So in the top left, you're targeting one sort of target, and the bottom right, you're targeting another. And then in the center point, that orange highlighted partition is a double positive, as you'll see as we work through the slides. So it's positive for both targets. And then again, all the software has to do is just count positive FAM plus FAM hex positive over negative, and then it will be able to give you a copies per microliter. The software just automatically tells you the copies per microliter. Okay, so up until that point, we've basically been just describing the technology for everyone, just so that they can have sort of a good understanding, a good sort of base background. But now we're going to look at the sort of the benefits of the technology, especially when we're comparing to real-time PCR. So if we look at this slide here, the first key benefit we're discussing in this slide is your absolute quantification. So in virus quantification and detection, it's really critical for effectively identifying, monitoring, and mitigating your viruses. So sort of viral infections, can ebb and flow throughout the seasons, and the population spreads and changes sometimes can result in a drastic change in an outbreak. So during such an outbreak, it becomes really important to understand and manage the spread of that and treatment of that virus. So sensitivity and specificity are really important when you're looking at responding effectively. However, when you look at RNA viruses, maybe things like Ebola and Zika, they have high variability, and they need to be detected in often what is very high backgrounds. So your DNA to RNA levels are you've got very low levels of RNA over DNA. So ddPCR is a really useful tool for something like this, for viral detection, because it offers quite a lot of advantages over qPCR alone. For example, it enables accurate quantification without any standards. It can verify the accuracy of a real-time PCR assay for cases where the virus contains sequence variants, which is common in RNA viruses. And ultimately, for low-level quantification, digital PCR is just going to provide easy and accurate results with a much higher confidence level. So if you look at the top, you can see traditional real-time PCR standard curve. And obviously, that this requires quite a lot of extensive validation. Because of that, looking at it at the exponential phase, meaning the data can often look a little bit overwhelming at first. But if you just consider the key parameters that tell us how good or bad your assay is working, you'll see On the left side of that sort of top chart, the amplification curves are color-coded for each of your gradient temperatures. That's what the different sort of, that's the assay optimization step. And you can see at a quick glance, we can see that the blue and the dark green curves have sort of the best sort of sigmoidal shape. So that's that S shape, and appear to cross the threshold earlier than some of the other temperatures, like your orange and pink, which are struggling. And if you look at the standard curve for each of those, you can see for each of those temperatures, we see that the lowest annealing extending temperatures have their best efficiency. How do we know that? Well, they have an R squared of around 1. So at that phase, there's a doubling, and at that temperature as well. So it does look complicated, but it's not too difficult to work with. Now, if you look at the same assay when run in ddPCR, there's no standard curves or standards required. So it's time saving factor is massive, as well as a cost saving, running all the assay optimization and running the cost of providing all those standards. You just have a provided absolute quantification of your unknown samples. So in the case here, we're looking at the viral load of Zika virus, and you can see it's across 5 10-fold dilutions. And you can see how accurate the assay is across those 5 dilution series. You can see it's going from about 5,800 copies per microliter to around 520 to around 52.6. And you can see it goes all the way down to 0.48. So very little variation between your samples across the run. And you can also see how well the assay is optimized on ddPCR. You can see on the right there's a gradient was run just like you can do on real-time PCR. And you can see we ran these samples from 55 to 65 degrees across the wells. And in A and H, you can see really consistent results around 1,050 copies per microliter across all the wells. So the effect of assay efficiency is much less noticeable when compared to real-time PCR. Now, looking at the second key benefit, we talk about this enhanced sensitivity of ddPCR over qPCR. So in this case study, we can look at HIV sort of cure research. So in populations with high HIV burden, there is a lack of accurate assays that are able to quantify the replication competent reservoir across the various HIV subtypes. Now, the HIV reservoir contains sort of latently infected cells, and they can persist even during suppressive ART treatments. So cells infected with HIV carry this proviral DNA copy of the HIV genome, and that copy is stably integrated into the host cell's chromosome. And this process of generating this proviral DNA through reverse transcription is sort of often error-prone, and most integrated proviruses contain sort of mutations, insertions, deletions, rendering them often unable to produce new infectious virus. So this means that the remaining cells that contain these full-length intact proviral DNAs can produce replication-competent viruses, and are they kind of the source of what is termed viral rebound upon treatment, ending treatment, and are a major, obviously, barrier to cure. So for many years, what's sort of been the gold standard was the quantitative viral outgrowth assay, and they rely on stimulation and culture of replication competent viruses. However, they're quite labor intensive, it's very low throughput, and they often underestimate the amount of replication competent reservoir that's left in there. However, intact proviral DNA assays detect multiple targets across the HIV genome. And in this case, you can use a multiplex ddPCR assay that are much higher throughput than traditional qPCR assays and other full-length sequencing reads. And we can show that across various subtypes, it works on A, B, C, D, A, and E, and you can detect up to a single copy of intact proviral DNA. So in this case study, the study was suggesting that early ART treatments in infants may alter the establishment and long-term persistence of HIV infection. So they needed a really sensitive test to evaluate the persistence of HIV infection. in the child through the age of 30 months during the therapy, and even after the therapy was discontinued. So they used ddPCR for this case, where they checked proviral DNA in 2 long-term repeat circles in PBMCs and also in CD4 positive T cells. So in this case study, an infant born to a woman with HIV type 1, they began treatments on ART for after just 30 hours after birth, and the ART treatment was continued until after the criteria was met. The therapy was then discontinued at around 18 months of age, and the level of proviral DNA in these PBMCs was assessed by clinical assays, including ddPCR, and they used these sort of to detect these levels and comparing them to qPCR assays as well. And their sort of take home from the study was ultrasensitive. ddPCR was an ultrasensitive test showing very low and measurable HIV DNA at 24 and 26 months that was not able to be detected by real-time PCR testing. So highlighting that much higher level of sensitivity when compared. And in this case, there was obviously just 3 types of tests done to compare. There was a plasma viral load, which detected a single copy of RNA. There was a co-culture assay, which could not find any replicant-competent viruses. And then there was the ddPCR assay, which can find 37 copies of HIV DNA in the PBMCs after 24 months, so up to 26 months still having detection. And then the third benefit of ddPCR over qPCR is tolerance to inhibitors, so inhibition tolerance. Now, in this particular study, they were evaluating the potential of ddPCR for accurate quantification of a waterborne virus at low concentration in environmental samples. So RNA was isolated from rotavirus and a variable in environmental samples, and they were set up as control experiments. So they compared these to qPCR. ddPCR showed a lower limit of detection than real-time PCR, being able to detect the presence of viral particles at less than 10 copies per microliter in reaction. And both ddPCR and real-time PCR could quantify down to 10 copies at the lower end. Although real-time PCR and ddPCR showed similar sensitivity and linear correlation, ddPCR surpassed qPCR on measurement repeatability, so the precision, and far outperformed real-time PCR in the lower part of the dynamic range. And what's really interesting is if you look at the samples that they ran, if they ran it in sort of milli-Q grade water, real-time PCR easily detected the spiked sample at a consistent CV and a higher copy number. But once exposed to a difficult sample, such as from a river, where there's a lot of inhibitors present, the copy number greatly dropped and the variation between replicates increased. This highlights the much larger tolerance to inhibitors that can sort of be attributed to using an endpoint PCR method rather than an exponential method. And the author stated, the results demonstrate the higher precision and repeatability of this ddPCR assays for quantifying waterborne viruses at low concentration in most samples routinely analyzed for water quality. So you can see in the slide, much greater repeatability in the copies due to much higher sensitivity, reduced sensitivity rather to inhibitors. And you can see the CV is much more stable. And when compared to qPCR, much higher CV values and very varying sort of copies determined as well. And then the fourth and final benefit that we're going to look at this afternoon. In this case, we're looking at BRAF. So cancer is obviously still a leading cause of death. That's sort of plaguing many populations, and scientists obviously finding a lot of genetic changes, mutations that are causing and driving these cancers. One of the most important ones is BRAF, which helps control cell growth. So mutations in BRAF can make these cancer cells continue to grow and spread at really fast rates. And there's a lot of BRAF mutations, I think over 200. but especially in things like melanoma, colorectal, and lung cancer. But one of the most common mutations is BRAF V600E. And we have drugs that target the specific mutation. And these drugs do improve patient outcomes. But obviously, you need to make sure that your patient does have this mutation and in what quantity, etc. So to normally monitor this, doctors would take a small piece of a tumor by doing an operation and a solid biopsy to diagnose this cancer. However, with this, it's quite invasive and it might not show the full picture of all the different cancer cells within the tumor. So what is a popular alternative starting to develop is this liquid biopsy. So it's a newer approach, and it involves just testing fluids like blood and saliva. And these fluids often contain circulating tumor DNAs, ctDNA, released by these cancer cells. So when analyzing these sort of ctDNA, it reveals much more information about the cancer itself, such as how it's responding to treatment, the stage, the patient's prognosis, and if any cancer remains after treatment. It can also provide a much more complete picture of the tumor's makeup because DNA from all parts of the tumor are found in these sort of fluids. However, current detection methods show that we do need a method that's more reliable and they can be improved. So normally traditional methods are something like real-time PCR, Sanger sequencing, and next-gen sequencing. And they're used to find these mutations in both tumor tissue and liquid biopsies. However, like we stated, there is a need for a faster, more sensitive and comprehensive method to detect these mutations. So in this study, they compared real-time PCR and ddPCR in liquid biopsy samples, and the images highlighting the difference precision in terms of the coefficient of variation between these three methods using the different target inputs. And they concluded among the technologies assayed in this manuscript, the ddPCR process is robust and shows clinical sensitivity, specificity, and high reproducibility for the profiling of actionable mutations present in ctDNA. Thus, the use of ddPCR can significantly improve patient diagnosis and treatment, as well as administration. Just in addition to this, one of the, so that's obviously discussing mostly to do with precision, but if we look at the reproducibility of ddPCR, we can see this is really one of the biggest strengths of the system. You can see the precision when counting of input templates on this BRAF V600 assay. So in this case, it was 3 different users, 3 different instruments at 3 different locations. And you can see that they produced almost near identical results across the whole range from 1% to 0.01% detection limit. And this is a really good benefit of ddPCR when compared to qPCR is the reproducibility between instruments and between different users across different locations as well. So those are just really the 4 main sort of highlights that we wanted to touch base on about sort of ddPCR. But keep in mind that ddPCR is really a broad sort of application. Anything you can run on real-time PCR and a lot more can be run on ddPCR. If you look at this slide, it's really focusing on the challenging applications that ddPCR can support. So we look at something like wastewater detection. That's because of the inhibition that's common in wastewater testing. We've got viral load and viral titer, because you don't have to use standards and it's not a relative method, much more accurate. You've got copy number variation. Copy number variation is obviously not such a problem for small copy number changes, but when looking at large copy number variation changes, your real-time PCR can no longer accurately determine the differences because you're looking at a doubling between each time. So if you're working between cycle 32 and cycle 33, it's hard to determine if you are accurately determining copy number when going between such high copy number states. Then you can also look at genome edit detection, microsatellite instability, which is a measure of sort of change in your cell regulation, and then mutation detection, like we touched on sort of with liquid biopsies and in tumor samples, really are some of the key benefits or challenging applications that ddPCR sort of thrives in. So yeah, just a brief overview. I'd love to hear if you have any questions or if you want to share anything about anything that you might have come across as well.
Thank you, Luke. Very informative presentation. Even I learned something new every time. So thank you very much and have a lovely day, everyone.
Thanks. Thank you, Michelle. Thank you, everyone.